Good afternoon, everyone. Afternoon here, at least. It's my pleasure to introduce Dr. Karl Broman, Professor of Biostatistics at the University of Wisconsin, Madison. Karl is a longtime friend and collaborator of many of us in this P30, and we are really delighted to have him here. Karl got his bachelor's from the University of Wisconsin, Milwaukee in mathematics, followed by a PhD in statistics from the University of California, Berkeley. He worked as a postdoctoral fellow at the Marshfield Medical Research Foundation in Wisconsin, and was a faculty member at Johns Hopkins University in biostatistics before he joined University of Wisconsin-Madison. Karl has made many important contributions to quantitative genetics, beginning with his early work on recombination rates in different species, and followed up by a kind of long-term work on genetic mapping in experimental crosses. And perhaps his most enduring contributions have been his work on the eQTL and eQTL2 software, and is very important and groundbreaking paper on the genomes of recombinant inbred lines, which were really very helpful for a lot of people who are working to build those resources. I think Karl has done many things, but if I have to find a unifying theme to his work, I would say that he has really taken a fresh, rigorous, and kind of a critical look at bread and butter issues for geneticists and quantitative geneticists. I think that if there's a theme, it is that he often looks at things that nobody else notices, and today's talk is one of those things that a lot of people face, but maybe nobody kind of devotes a critical eye to that. And without further ado, I would invite Karl to talk about sample mix-ups today. Thank you, Karl.
Thanks, Shaunak, and thanks so much for having me as part of this series. It's really been a delight to hear these this series of practical talks that you've assembled. And I think maybe one of the features of the pandemic that this in-person course got turned into an online experience for all of us, and expanded to this is the 17th talk, I think. So I'm going to talk today about sample mix-ups, and I'm not going to show any code. I'm going to focus on the kind of the principles that I use to try to identify sample mix-ups in expression genetic data. Feel free to stop me as I go along, or I'll stick around until 1:30, or you can find me on Twitter. You can find everything about me on GitHub. You can find the slides for every talk I've given since like 1996 on my website. The slides for this particular talk you can find here. So systems genetics is a lot about trying to find associations between different sets of data. DNA-based genotypes, gene expression data, clinical phenotypes, proteomics data, microbiome data, all that. You have groups of measurements on basically on the same individuals, and you're trying to find associations between them in order to learn about the underlying mechanism of human disease and other things. If the labels on the samples get mixed up, it will reduce those associations and interfere with our ability to learn about mechanism. The most extreme case of this for me was this one of the first studies I was involved in here at the University of Wisconsin. A large mouse intercross with 500 mice and a high density array-based genotypes, high density in 2007, like 3,000 markers, and array-based gene expression data on 6 different tissues. And in the process of analyzing these data, we ultimately discovered that there was almost 20% of the DNA samples that were mislabeled. The DNA samples were arranged in these 8x12 plates, and a dot here indicates that the correct sample was in the correct location, but the arrows are pointing from where a sample should have been to where it actually ended up. So there are some odd sets of sample mislabelings, but the bulk of the problems were a series of off by 1 and off by 2 errors for half of each of 2 plates, so that almost 100 out of the 200 samples had the wrong labels on them. And we didn't discover this until we'd been studying the data for quite some time. And those were not the only mix-ups in that data set. As I said, we had array-based gene expression data on 6 different tissues, and there were sample mix-ups in each of those panels of data. We didn't find any mix-ups between tissues, but within each tissue there were some subjects that were mislabeled. And I think the first paper that I've seen on this topic was this Wester et al. Paper in Bioinformatics in 2011. It came out at almost exactly the same time as I was trying to work out this problem. But they had maybe the first formal method for trying to identify mix-ups in eQTL type data. They applied it to a range of publicly available data sets. Getting access to the data behind many of these papers can be difficult, but there was one data set, one paper where 20% of the samples were mixed up, only 100 samples or so, but these have quite a big impact on their ability to identify eQTL, and correcting the mix-ups makes you get much better use of your data when the samples are correctly labeled. So my outline for today is to talk about how we go about finding these things. I'm going to start by just looking at kind of the part of the process that I use in doing data diagnostics. I'll first look for sample duplicates. I'll try to verify the sex of the samples, and then I'll look for sample mix-ups. And I'm going to use a couple of different data sets, and I won't tell you what they are. I have a tutorial on this topic where you'll be able to see the actual code that I'm using, but I have not been able to post it yet because I'm still waiting on the correction to a paper being published, or a correction being published to a paper whose data I'm using. You can maybe guess what the data sets are, but I'd rather not talk about the particular data, try to infer the data sets today. Let's just talk about principles. But feel free to ask me anything. But first, always look at the pattern of missing data. Before you do anything when I get new data, the first thing I'm going to look at is missing data, because it turns out that missing genotypes, missing data is very often a good indication of problems with samples. And they, a problem sample will show up, will kind of interfere with everything else you're trying to look at. So like the first thing to do is look for missing data to try to identify and eliminate problem samples. So for example, here, this is data on diversity outbred mice, about 500 mice. And I just take, available SNP data and count what is the proportion of missing genotypes for each sample. And the bulk of them are missing no more than 5% or so, but there are a couple samples that are missing more than 50%. And here we see 7 samples that are missing more than 10%. And I would say these top 5 are clearly just bad DNA that we might as well just omit and not worry about further. These 2 probably also are not going to be recoverable, but we could probably leave them in. These data were generated in 5 batches, and you can see there are pretty big batch differences, which is very common in all these high throughput genomics or omics studies. So the data sets I'm working with are these diversity outbred mice. They're very similar to heterogeneous stock in starting out with 8 strains and doing random mating with large populations for many generations. Diversity outbred mice are a little different because it was like they were starting to make recombinant inbred lines from 8 founders, and then partway through they decided to take those and start mixing them and creating this outbred stock. But the way that, their genomes are largely the same as heterogeneous stock, and that the data that I'm going to be looking at is of this form. But I think that the principles we're using would apply to any kind of experimental cross, I think, and likely could be applied without much change in human studies. Although an advantage of diversity outbreds and heterogeneous stock is that we don't really have many or any rare alleles, that the most rare alleles are not really rare at all. And that is a big advantage for a lot of things. So having maybe tossed the samples that have 50% missing data, the next thing I usually do is look for sample duplicates of just take the genotype data and ask, are there pairs of individuals that have basically the same genotype data? So just take the SNP data and count proportion of matching SNPs across, 80,000 or 100,000 or whatever, 10 million SNPs you're working with now. And in this kind of project, you'll find that, the bulk of the samples are, smack in the middle, say. And in these diversity outbred mice, we'll occasionally see groups of pairs of samples that are, sharing quite a bit more. That these are almost all likely siblings. There weren't supposed to be siblings in this project, but it seems to me that there are siblings here. There's a group of pairs of samples that are sharing less. All of those are pairs that involve one of these 2 samples that was missing a ton of data. Leaving those in, I get this little blip down here that are just like, these 2 samples are just bad. What I'm looking for here are samples that are way out here near 100%. If you think 1% missing data, or 1% genotyping errors, then 2 samples that are really identical repeated, you should see 2% difference. If the error rate in SNPs is often less than 1 in 1,000, and so you'll have very close to 99% matching at 2 identical samples. I don't see any duplicates in this particular project, but I would say most of the time I see sample duplicates like this, or pairs of individuals with identical data. It's hard to distinguish that well, it's impossible to distinguish that from monozygotic twins. And no one's ever really, I still don't really know whether monozygotic twins happen in mice or rats. I'm interested in that question. And if anybody knows, please let me know, because I'm curious. I presume that if monozygotic twins happen, we'd see it more often. But we definitely see duplicates now and then, and we just look at percent matching genotypes with the raw SNP data, and you're looking for guys out there. You could maybe, with gene expression data, you could maybe look for the same kind of thing. Do I have two arrays that really look very with really similar gene expression data? Here's a batch of I don't know, maybe 500 RNA-seq experiments, 500 samples, and I just looked at take all genes, just look at the correlation between samples, and centered around 0, a lump that's to the right and a lump that's to the left of 0, a little bit positive, a little bit negative. These are same-sex pairs and opposite-sex pairs. None of the correlations get very high. You might want to do something to try to eliminate unexpressed genes and just focus on the correlation between expressed genes. It's kind of tricky to do. I'm pretty sure that but nothing I could do would really work. And I think that if I had two duplicates, it would show up here at being unusually high correlation. If I adjust for sex, that the whole distribution gets centered closer around 0. I illuminate these two lumps, but none of the correlations are still very high. You got some that are like approaching 0.75, but I don't think any of these are really duplicates.
Karl, is that for liver? Is that liver?
I think it is liver.
Yeah, because it's way more that you could almost begin to see, sexual dimorphism in the official sense of two modes. I've never seen anything like that in brain, any tissue, even hypothalamus. That's cool.
So the tissue here has a huge sex difference, which can be useful, as we'll see in a moment. I mean, this data set also had proteomics data, and you could do, I think you should be able to do the same sort of thing. If you have a batch of proteomics data, you should look to see if there are, pairs of samples that are more similar than they should be, because that would indicate some kind of duplicate. So the next thing I'll look at is try to verify sex. Male and female samples. The old school approach was to look at heterozygosity on the X chromosome. If you saw heterozygosity on the X chromosome, that was a female sample. If you didn't see it, then it was either male or just a female that happened to be homozygous on the whole X chromosome. But now with either, I think, SNP array data or with the different kinds of sequence-based genotypes, I would look for dosage. It is much more informative about sex. Look at the average intensity of X chromosome SNPs and plot it against the average intensity of Y chromosome SNPs, and it should separate really nicely a group of males and a group of females. That females shouldn't have a Y chromosome, and they'll have 2 X chromosomes, and males will have just 1 X chromosome and a Y chromosome. And you can, if you see an annotated to be female sample up here with the males, or an annotated to be male sample down here with the females, then it would look like the sex annotation was wrong, and you'd want to figure out why. Here there are a couple samples that have low dosage of X and no dosage of Y, so that those are maybe XO females. Occasionally you'll see a dot up here. If you have some samples that have problematic samples with a lot of missing data, we'll kind of be all over the place in this sort of figure. But this, just looking at the intensity or the total reads mapping to the X and the Y chromosome makes it super easy to identify the sex of samples. Seems like you should be able to do the same thing with gene expression. Maybe I have an advantage here that it's in liver, where there's a big sex difference in gene expression across the board. I mean, you might think, well, let's think about some genes where you really expect to see a sex difference. Or my approach tends to be not really think, but just do a whole bunch of t-tests and identify the genes that show sex difference and then look at those. And if I do that, well, the gene that I would expect to show big sex difference exists, related to exon activation. That does show, high expression in female, low expression in males. There are, some samples that are down here then that maybe shouldn't be, and some samples that are up here that maybe shouldn't be. I have lots of genes that are showing big sex differences, some where the, more highly expressed in male than female. So one approach would be to just identify the top 100 genes in terms of sex difference and say, calculate the average for males and the average for females at each of these genes. And then say for every one of my samples, which is it closest to? Is it closer to the male averages or the female averages? So that's what I did in this figure, is I plotted the distance from the female average and the distance from the male average for this selected set of 100 genes. And you see, the females are over here and the males are over here, and there's a smear of individuals in between that are not really doing quite what I want. But there are, there's a female in the male ball and a few males in the female ball that would be worth looking for. I mean, looking at to see what's going on. I think the sex difference in gene expression is a useful tool to to help you to identify whether some of your samples have been mislabeled as something that has a different sex. You could
just to add there, Karl, if you had the body weight of the animal, it's also a kind of nice reality check that's pretty easy to get hold of.
Right. And very good. I mean, other kinds of simple Mendelian traits like coat color, you can use and compare to genotype at the location where the coat color is mapping to, and that can help you to identify some sample, mislabelings too. We can do the same thing with proteins. In this data set, there are a bunch of proteins that show strong sex differences, not as strong as the gene in the gene expression data. But I make the same sort of plot, and you get, a good separation between males and females doing something really simple, with, some males or some females stuck into the male group. And, a smear of individuals in between where I'm maybe going to be less confident about what sex the sample might have. But let's now turn to the meat of the topic, which is to use this, large high dimensional data to try to identify sample mix-ups. First, so this particular data set, I have, RNA gene expression and proteomics data, and I can ask, do the sample labels in those two data sets look to correspond or not? In in other studies, you might have gene expression data on multiple tissues, and you can ask, do the sample identities in those multiple tissues seem to line up the way you'd expect? And the basic approach is, I have this rectangle of gene expression data of, genes across the genome and their expression in a set of mRNA samples. And I have this rectangle of proteomics data, protein samples measured, proteins by mass spec across, a set of proteins. And some of these rows are supposed to correspond. And I want to ask, how, do they actually correspond? The first thing that I do is look for pairs of a gene and a protein where expression of the gene is highly correlated with the, the protein content. Now, I could really, think carefully about which ones I expect to be the same, like, the protein product corresponding to a particular gene. I could look at the annotations and focus on those pairs. Or I could I'm going to just look at all pairs of genes and proteins, or for every protein, find the most correlated gene. And then I'm going to focus on a subset of genes that are highly correlated with the corresponding columns of proteins. So pick the top 100 proteins, and in terms of having a highly correlated gene. And then with that subset of highly correlated variable pairs, I'm going to look at how similar are different, of an mRNA sample and a protein sample, of look at, say, just like the correlation between the set of gene expressions here and the set of protein levels here, and use that to form a correlation matrix. How correlated are the protein samples to each mRNA sample when I focus on this set of correlated pairs, and use that as a measure of similarity between protein samples and mRNA samples. Hoping that the samples with a sample, the mRNA sample and the protein sample that came from the same individual will be more correlated than to any other. With this particular data set, there were, 7,000 proteins. If for each of the 7,000 proteins, I ask, what is the most correlated gene? The most correlated gene in terms of gene expression was, most proteins had a correlated gene, were correlated with some gene at about 0.3. And there were some that were way out here near 0.8. And the top 3 most correlated protein with a gene were all cases where it was a gene and its protein product. I chose to adjust for sex in doing this. Not sure that's the right way to do it, but it makes eliminating the huge sex effects that we have in gene expression and protein here makes the plots easier to follow, easier to understand. And so I chose to do that. I'm not totally sure that that's the best approach. But so the idea is, for each protein, find a correlated gene, find the most correlated gene, and then let's stick with, say, the top 100 proteins and their correlated genes. And then for every protein mRNA sample, look at the correlation across that 100 genes and form this similarity matrix. 500 or so RNA samples and just under 200 protein samples. Black here means similar and white means not similar. There are some black lines, diagonal lines here. Those are the same sample ID. This like checkerboard pattern throughout that, I think that is some set of batch effects. So again, I kind of adjusted for sex before doing this. And if I had not adjusted for sex, you would see really two big, well, a checkerboard with two 4 squares, because the sex difference between these samples is enormous. So if I then separate, I take that similarity matrix and I separate the set of pairs that are, have the same label from the set of pairs that have different labels, I should really see the, samples with the same label have high similarity and samples with different labels have low similarity. And I'm hoping to have no overlap between these two distributions. But you do also see that samples that have the same label, some of them are really low correlation. They're like draws from this not actually related distribution. And down here, you can kind of see that there are some samples with different labels that are highly correlated with each other. So and a simple summary to take is kind of think of that that similarity matrix, and for each row, each protein, find the biggest similarity value in that row and plot that against the RNA sample that has the same label. So it's sort of self-similarity and the most similar sample. You plot those against each other, you'll find that most of the samples, for most of the protein samples, the RNA sample with the highest similarity is the sample with the same label. But there are some samples where that the protein sample and the corresponding RNA sample are not all that similar, and there's some other sample that's really quite similar. So these are all mislabeled samples where there's another sample that it looks like it's what it should be. And these down here at the bottom are a few samples where they look to be mislabeled. They're not highly, the protein and the corresponding RNA samples don't look similar, but I can't find another RNA sample that really matches that protein sample.
That's a cool plot. I love that.
So we can look, let's pick a few of these and look more closely at them. So protein sample M348. If I take the similarity matrix and just plot the similarity of this protein sample M348 against the similarity to all the other proteins, to all of the RNA samples, I find it's actually closest to M410 and quite a bit closer to the RNA sample M410 than to any other sample. And the orange dot here is the correspondingly labeled sample. Whereas down here at the bottom, I have the M410 protein sample, plot its similarity to every other sample, every RNA sample, and it's most similar to M348. So those two samples, it looks like there was a sample swap. The proteins or the RNA samples labeled M348 and M410, one of the two pairs there was a sample swap in the labels. And this pair of samples is the same way. F371 is most similar to M349. There are a couple other samples that are, kind of similar too, but it's pretty far above all the rest of the samples. Whereas these two samples here are M386 is most similar to itself, to the RNA sample with the same label, and F352 is most similar to the RNA sample with the same label. And so in looking at this data set, 200 or so protein samples, 500 or so RNA samples, there were, going back one slide, about 9 pairs that were all ended up looking like this, of a sample swap in labels, two samples that got swapped. We don't know whether these swaps happened in the protein data or in the RNA data or some of each. We need some other information to help us to figure that out. It can be that the identities, figuring out where the samples sat next to each other in the freezer on plates, when the samples were managed, could help. You notice M348, M349, those 2 samples with numbers right next to each other, both being involved in this, is the kind of incriminating information that you'd be interested in looking at. So that's, RNA sample and protein sample. Focus on a subset of the variables that are highly correlated between the 2 groups, and then look for correlations between rows, and to try to find samples that are not very correlated to the correspondingly labeled sample, but is more correlated to some other sample. We'll do the same sort of thing with DNA and each of the other 2 batches. We'll start by looking at the RNA gene expression data. So again, I have a set of genotype data, a set of gene expression data. The first thing I'm going to do is for each gene in the gene expression data, I'm going to map to try to find an eQTL. And then I'm going to focus on a set of genes that have really strong eQTL. So reduce the data set down to the portion of the gene expression columns that have some really highly correlated genotype at some spot in the genome. And, so pick genotype and expression pairs where there's a really big similarity, or where there's a big association between what genotype you have at the eQTL and your expression at that gene. And then I'm going to use these data to create predicted expression data for each DNA sample and each of these genes, which maybe I'm not supposed to call genes. Get what would I predict its expression to be given its genotype at the eQTL. It's basically here at an eQTL that has a strong effect on the expression of this gene. Just take the average expression for each of the 3 genotype groups and use that as the predicted expression for that gene to tell me the genotype at this eQTL for each sample. And I'm going to just pick the average of these, the average of these, or the average of these as its predicted expression. And then for every DNA sample, every RNA sample, I'm going to just calculate, how similar are the predicted expression values to the observed expression values for this subset of genes whose expression is highly correlated with, has a strong eQTL. And the thing that I'm going to do here is just take the RMS difference take the difference between the 2 vectors, square them, take the average, and then take the square root. And use that as a distance matrix where large values here are close, where a DNA sample is very similar to this RNA sample and vice versa. So for these particular data, the 20,000 or so whatever genes, this is the distribution of the genome-wide maximum log score for each of those genes. typically not very strong, but there are a whole bunch of genes with really strong associations, including 100 that have an eQTL with a LOD score of, about 140 and more. So I'm going to focus on those 100, the top 100 in terms of they have a super strong eQTL. And then I, use the genotype expression association to get predicted expression data for each of my DNA samples. For this project, I have like, 850 DNA samples with my 500 RNA samples. And I have predicted expression data for each of these DNA samples. And I say, how similar is it to the RNA expression data, the observed expression data, by just taking RMS difference between the two things, or sum of squared residuals, but with an average in there. Some, striking diagonal lines in there, which are, I can see some of these look to be the same sample. So here, black is close and white is far away. And I do the same thing that I did before. I could separate the samples, the DNA samples, RNA samples with the same label, and look at the histogram of their distances, and the DNA samples, RNA samples with different labels, and look at the distribution of their distances. And here they look, there's almost complete separation of the two distribution. And there's really no self samples that have large distance. There are some non-self samples that have kind of small distance, sort of overlapping there. But then if I do the same trick I did before of take my distance matrix, and for each RNA sample, find the closest DNA sample and compare it to the sort of the distance from itself. Here, this is clean. This is what we're hoping for. There's all of the samples for every single row. The closest sample is the one with the corresponding label. And if let's just pick out those same 6 samples that we looked at before. So take RNA sample 348 and then compare how close is it to every sample, each of the 800 and so DNA samples. The closest one is itself, and much closer to itself than to any other sample. This one, M386, there's another sample that's kind of unusually close to it, but really these are all as clean as clean could be. So in this data set, we see really no mix-ups in the labels of the RNA. So the last bit is to ask, so okay, now let's finish off the triad and compare the DNA to the protein. We're going to do exactly the same thing we did with the RNA of for my, 7,000 or so proteins. For each of them, I map protein level, do a QTL analysis, and find the strongest pQTL. And then I focus on I'll just focus on the top 100 proteins in terms of that have a really strong pQTL. If this particular protein and its corresponding strongest pQTL has some association, I use that to get predicted protein levels for every DNA sample. And then I compare each DNA sample to each protein sample, and I get this sort of distance matrix. This is exactly the same picture I showed before, just changing expression to gene expression to protein. Here I have just 200 proteomic samples. The LOD scores are all smaller, partly because of that. Just the sample size is half, but there are 100 or so proteins that have a pQTL with a log score that's, about 30 or above. You compare that to 150 and it's like, wow, these are small, but compare that to like anything else we do. 30, I mean, these are really strong associations. And then compare the 200 protein samples to the 800 or so DNA samples, and you see, these strong black lines, which are the samples with the same label. Do the same plot of separate the protein DNA pairs with the same label and the protein DNA pairs with different labels, and a nice separation between the two distributions, except again, here we see a bunch of pairs with the same label, but are distanced from themselves, and a bunch of pairs down here with different labels that are really similar to each other. Make that same plot again, take my distance matrix, and for each protein sample, each row, find the closest value and find the distance at the corresponding value. And you see, most of the samples, the closest sample is itself, but there's this group of samples out here where they are not particularly close to themselves, but they're close to something else. And so these are all, again, the mislabeled samples. Some of these have pretty high distance to the closest other thing, so maybe not necessarily, we can't necessarily identify what is the corresponding sample here. If we go and look at the selected set of 6 samples I'd shown before, we say that the same sample swaps that we saw when we compared RNA to protein, that M348, M410, they look to be swapped between the two. And M349, F371, they look to be swapped between the two. So having the protein, the gene expression, and the DNA, the 3 of them together, shows that the problem in this data set looks to be in the protein sample, not in the DNA sample, not in the RNA sample, it's in the proteins. So in summary, stuff like this shouldn't happen. Whatever procedures we're using in the lab that allow 20% of the DNA samples to get mislabeled, we should stop doing that. But if it does happen, with this, high throughput phenotype type data, we should find it. We can find it. It's not, it's kind of fun to find it. So so before you do anything else, give a quick check to, can you find sample mix-ups in your expression, protein, other high throughput data? And basically, anytime you have multiple data sets whose rows are supposed to correspond, you should check to see, do they correspond? The main approach I used was find highly correlated columns between the two data sets, and then use that to measure similarity between rows. And showed us that, when comparing the DNA and RNA here, it was perfectly clean. The rows looked to be correctly labeled in both sets. Whereas comparing either DNA to protein or RNA to protein, we found some clear evidence that there were mix-ups. I mean, what's comforting with this, nicely measured RNA and protein data, it looks like we have enough information in the genome to be able to, there's enough signal in these data sets that we can correct any problems that might occur. So I mentioned at the beginning this paper from 2015 of mine, and also this top paper by Westra on that. They both, 2 different methods of doing this. I think I'm at this point leaning more towards the method that Westra used rather than the method that I developed there. This other paper by Lynch et al. In PLOS One in 2012, it is doing the same sort of thing in human GWAS data, and has some nice comments about experimental design considerations in this sort of project. I'll also point you towards this paper from a couple of years ago on cleaning genotype data and diversity outbred mice that has more sort of my data diagnostics methods. But thank you all for having me. The slides I've presented are online, or you can find me online, or all my code on GitHub, the source for these slides on GitHub, and you can find me on Twitter.
Great stuff, Karl. Great stuff. There's a question from Steve that I think is perfect given your next to last slide, or where he's, Steve, you want to ask your question? We can't hear you. Okay, now.
Yeah, I just got unmuted. So now I will. Yeah, my question is, in terms of missing data, where you have bad samples, low quality DNA, RNA, whatever, or where you have sample mix-ups, what's the best way to anticipate that at the sample preparations stage, at the front end of it, in an attempt to, maintain the power of the experiment that you're going to run?
What do you think, Steve? I mean, my experience in the lab is theoretical.
So what I think is run more samples, but each sample costs another, we got a budget, man.
So my response to that would be, if you're running a mouse genotyping service like Transnetics for a company like HHMI to get mouse genotypes right, and you want to maintain that business, which brings in about $20 million a year from all kinds of institutions, you cannot afford these QC steps. You have to work really, really hard on the QA. You can't afford the screw ups. But those of us in academia, we don't even know what QA stands for. So if there are 30 people on the webinar now, and it'd be fun to do a poll and say, who knows what the A in QA stands for? We all see the QC, it stands for quality control, but you want to avoid the control if you possibly can. And so the assurance step is what the big boys have to cope with if they're trying to make money. And I don't think we're good at it. I've been doing this and screwing up now for 30 years, and I'm just so totally used to it. I do a QC. I don't even bother with the QA really anymore. I've just given up on controlling that.
But Karl's whole, sorry.
So
can I say, I mean, I constantly am telling the people in the lab how common these mistakes are and how costly and awful they are. And I think that's a cultural thing to try to make people understand that it's really easy to screw this up, and it's really serious, and it's really expensive. We have barcodes that they scan so nobody's writing, and we have chips in the mice and the rats so that it's harder to screw up who they are, and et cetera. And I spend a decent amount of time in the weeds trying to think of systems that are robust to these kinds of problems. You only have one tube open. When you're killing animals and collecting tissue, there can only ever be one sample tube open. It's the tube for that animal. As soon as you've got two tubes open, you can screw it up. And, so.
That's a good point. There are some good things that you said, Abraham. What is are these common knowledge, or these are things that you came up with? Yeah, what you just said, it looks pretty rigorous and systematic.
You know, I don't know how to answer that. I've heard Karl talk about the
can
story,
I
parts
just
of the story
interrupt for a
before,
second?
and it made an impression on me. And I've had my own experiences of screwing things up, and it made an impression on me.
So can I just interrupt for a second? Would you mind just systematically going through those steps? Because we're setting up to do, you know, a large scale eQTL experiment. I mean, in flies, so we can't chip them. I mean, we have the genomics data, but just if you wouldn't mind, just for my benefit.
I'd love to talk to you, Steve. I've got to be on another call in 3 minutes, so
Oh.
I'm probably not going to do it right now. But I'd love to do it, and I'd be happy to make it public. You know, I mean, there's
And make
Sure,
it a cell
sure.
paper, Abraham. We want to see it in cell or neuron.
Yeah, I'll touch base, but I'll also go back and listen to the recording.
yeah.
I mean, I think, you know, another point is having the people working with the samples really be connected to the project and care about the project. You know, as Abraham mentioned, like, you know, having the people involved really know the importance of their work. It makes a big difference. And that, you know, this particular project here, it was like the DNA samples went to another lab, which went to another lab. And the person that was actually working with them and probably, you know, like using a single channel pipette to move them from one format to another, like it was just a set of samples they had to work with probably. And that is maybe the worst situation of like, they don't really care that much about the project because they don't know what these samples are that they're working with.
Yeah, so being a newbie at all of this stuff, I have personally made the RNA samples, you know, in individual Eppendorf tubes, and then with a single channel pipette, put them into the 96 well plate that then goes to the sequencing center. So it's like, yeah, I mean, we have to figure out the automation steps to, just like you're suggesting.
There's a comment from Suzanne Mitchell. Do you want to comment or elaborate there?
Sure. I guess just quickly, I think it's really, really critical for the people in the lab to understand errors happen and not try to sort of breeze through it and be comfortable. And even admitting, oh, I think I might have screwed up there. I'm not sure, but let me write that down and annotate it. And then, you know, to go back and yes, you're going to do QC measures, but it also gives you a place to look. And also, I think when the staff are comfortable telling you that there may have been a problem, you can refine your procedures as well as knowing where to look.